I am very, very pleased to introduce you to Dr. Ilaria Capua. Dr. Capua directs her own research, but in addition, her lab is the FAO and OIE reference lab for avian influenza and Newcastle disease, and the OIE Collaborating Center for Diseases at the Human-Animal Interface. Dr. Capua developed the DIVA strategy, or differentiating infected from vaccinated animal strategy, which allows trade of poultry to continue even while actively fighting the spread of avian influenza. She has launched the global initiative on sharing all influenza data for virus sequences, which was a major step forward for research and for transparency. She has authored 150 plus peer-reviewed publications, and in 2007 she was among the winners of the Scientific American 50 Prize, and in 2008 she was included among Seed Magazine's Revolutionary Mind series. So please join me in welcoming Dr. Ilaria Capua, who will discuss managing the influenza matrix in line with the One Health vision.
So good afternoon, everyone, and thank you for being here and for eating your lunch while I am talking. I mean, I really appreciate that. I mean, you could be sitting somewhere else, so thanks a lot for that. I would like, of course, to thank the people that invited me here, the Center for Biosecurity. But I think that I'm really, really lucky because all the speakers that have spoken before me have actually set the scene for what I'm going to say. And a lot of the things that they have said are actually going to be picked up during my talk. And I've heard a lot about fragmentation and working together and finding solutions. And, you know, I think that it's time for us to show a way and see whether this can be done. And I think that influenza is actually one of the best examples with which we can work to show that One Health is more than a theoretical concept, but things can be put together to make it work for improved human health and animal health. So that was my take-home message, and now you can go and have your lunch somewhere else. Okay, so the first question I have for you is, do we think that influenza pandemics are something to worry about? And this is a very tricky question because it really depends who's sitting in front of you who will give you a completely different answer. I was at FAO last week, and I asked people to raise their hands, and of course, people living in the developed world are concerned about influenza pandemics. People living in the developing world are concerned about a lot of other diseases. So there might not be a correct answer, but I think that it is something we all need to worry about, and I hope that I'll be able to convince you. So what do we know about these viruses? Where do they come from? Well, they come from animals. I mean, this is the answer. The human influenza viruses all originate to a greater degree or a lesser degree from animals. And in fact, these viruses are perpetuated in wild birds, mainly. All influenza subtypes are naturally maintained in wild birds. From wild birds, they pass into what we would call intermediate hosts that are different species of animals in which they circulate and recirculate, and then they spill over into what we would call final hosts, which are humans, in which they cause pandemics, or they cause sporadic events of influenza known as zoonotic influenza. So you can see that these viruses are the perfect viruses that fit into the One Health concept because they infect so many different animal species. And although we like to think that we are different from animals, as human beings, I must tell you that viruses tend to look at receptors and not at whether we have a more developed brain or a soul, and therefore they don't really tend to make a lot of distinguish between us and other animals. So the influenza pandemics over the last 100 years, all of them have had an animal component. The Spanish flu appears to have originated from an avian virus. There are some papers that say that it might have passed through another intermediate host, but certainly it was a fully animal virus. The Asian flu of '57 was of avian and of swine origin. The Hong Kong flu of '68 picked up some avian genes onto a human influenza backbone, and the H1N1 pandemic of 2009 originated from pigs, as we know, and had genes originating from viruses of human and avian origin. So how does this pandemic influenza work? So I hope that I will be able to shed a little bit of light on how the zoonotic influenza works, and like H5N1, and what does that mean, and how pandemic influenza works. So these viruses circulate in animals, and in which they cause some disease, more severe, milder disease in a variety of animals. And what happens is occasionally these viruses knock on our door, and they knock on the door of human beings, and you have a sporadic event. And this is what has happened and is still happening with H5N1. This virus is by no means disappeared. It is still there, alive and kicking. But it causes only what we would define sporadic outbreaks. If these sporadic outbreaks, though, become more and more and repeated, the virus has the opportunity to adapt and to become what we would call pandemic, and then what we would call seasonal. And this is what happened with H1N1. The pandemic virus, it originated from pigs, it started knocking on our doors, on the human door, and it got adapted enough to take off and generate a pandemic. We do tend to forget that apart from human health implications, there are some very severe animal health implications. This is what highly pathogenic avian influenza looks like. It is one of the most devastating diseases there are in the animal kingdom. And it is causing a lot of problems to the livelihoods of rural villages and to the lowest income countries. And so when you're trying to manage a pandemic or the emergence of a pandemic virus, you never have to forget that there are some animal health implications in there as well that have a whole series of ramifications, including trade, including barriers to trade, the livelihood of the poorest components of society, and so on. And so it's a very complicated issue. Now, I think that we owe a tribute to H5N1. I think that a lot of people in this room owe a tribute to H5N1. I personally do, because I think that H5N1 was a real game changer. So just one slide on where are we. H5N1 is not gone. It is still endemic in 5 countries, and it is particularly severe in a couple of countries, including Egypt and Indonesia. The virus is an acrobat virus. It is capable of infecting many, many avian species, many mammalian species, including human beings. It is still causing 50% fatality in the human being it infects. And it has to be said that human beings are immunologically naive to this virus. So if this virus were to take off, it would most certainly create I think a catastrophe. And it is causing food security in developing countries, because as I showed you before, this virus actually kills animals. And since it kills animals, there are a lot of people in under-resourced countries that are suffering because of malnutrition, because this virus is killing their primary source of protein. And I would like to point out that everybody eats poultry. So there's no religious barrier or ethnical barrier to eating poultry. And poultry is extremely important, both as rural poultry, as scavenging poultry in many parts of the world. But H5N1 is, I think, as I said, is a game changer because it is the first animal virus that was mentioned by most political leaders of the world at the time. H5N1 was on the G8 agenda of 2006. And therefore, it has a legacy. And this is probably the most important thing that I believe we have to manage. Because as we heard earlier, there have been significant investments in capacity building. There is an increase in the number of veterinary and human laboratories that can diagnose this disease. We have contacts all over the world. We know who to phone, and people that are in the most remote areas of the world know how to get at an international reference lab. There have been expansion of areas of research, plus we have an unprecedented wealth of isolates that are coming from the animal kingdom and are coming from human patients that contain genetic information that is the key to understanding influenza infections better. And above all, it has provided an opportunity, which is to get OIE, FAO, and WHO to produce this document, which was produced at the interministerial meeting in 2008 in Sharm el-Sheikh, which is basically, how are we going to work together? How are we going to move this forward? So, what's next? How good are we at predicting emerging diseases? How good are we predicting the next threat? Well, I think that the H1N1 pandemic, which was certainly, let's say that our prediction capacities weren't very good because it came out of pigs. Everybody was looking in poultry in Southeast Asia, and the virus came out of pigs. And like, we weren't really prepared for this, and we weren't prepared you know, to face all the questions and all the criticisms that would have come. And this virus is actually a superstar virus because it is a real harlequin of the globalized environment. It has not only genes that come from viruses of different origin, but for the first time in history, it has two genes that come, so it has a combination of genes that come from two hemispheres. So this virus has genes coming from the Western Hemisphere and from the Eastern Hemisphere. And I would like to point out that actually the first case was, of course, recorded and identified in April 2009 in California, but there had been, the virus had been circulating at least for 2 months before in Mexico. And a question I have for you is, had we had those extra 2 months, would have our response been better than what it was? So, for the pandemic skepticals, was it a real pandemic? I don't know how many of you have seen that movie, My Big Fat Greek Wedding, where there's that guy that says, from the Greek. Well, you look from the Greek. Pandemic from the Greek means pan means all, and demos means people. So pandemic from the Greek does not mean that it's going to kill everyone. It means that it's going to infect all the people. And this is something that, you know, I think we need to keep clear in mind. And these slides I took off the WHO website because there's still a lot of pandemic skepticists. And just, if you look at, up there, there's the date. So they're taken every sort of month and a half. And this is what an animal virus can do over a year. And you tell me if this was not a pandemic from the Greek. So there you are. Those are the countries affected, and these are the number of deaths. July, end of July, 2009, September, November, January, April. So I think that we have an answer there that it was a pandemic. But I think that we have learned a lot of lessons. And personally, and as a member of the scientific community working on influenza viruses, I think that we've learned that given the tools we have just now, we have not been able to predict when the virus is going to emerge, where it's going to emerge, and what subtype it's going to be. Because those three questions were completely unanswered when this virus emerged. We can say that if you get a good virus which has a good engine, worldwide spread can occur within a few weeks. We can say that we were unable to predict how severe it was going to be and the age categories it was going to affect, or the ethnic susceptibility. And we have learned that vaccine was available after the epidemic peak, and therefore that the virus basically went faster than we did. And I mean, this is nobody's fault. These things happen, but I think that we should, learn from it. And I have to say, though, that before the pandemic emerged, my group did some work, and if you see the bottom statement in blue, we did some work suggesting that maybe we should be pointing more attention to viruses of the H1 and H3 subtype coming from animals rather than only to H5 and H7. And this was presented at a closed meeting, OAE, FAO, WHO. And it was actually rejected by Science, this paper, saying that, maybe we should look into H1s, and a couple of months later, that's where the virus came from. So anyway, I think that certainly in the veterinary community, there are areas of knowledge which can complement the existing areas of knowledge, since these viruses come from animals. So let us look into the future, and another question I have for you is, does it make sense to put influenza viruses in boxes? And you know why I say this? Because in the influenza world, until very recent times, until H5N1, and it's still very much like this, you ask somebody who does dog flu, do you know anything about swine flu? And they say, oh no, oh God, no. And you ask anybody who does avian flu, will you help me with something concerning dog flu? And they say, no, 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 that's none of my business. And it's been and this has been the case also very much between the human aspects of influenza and the veterinary aspects of influenza. But if you just think that these viruses all originate from birds and then they spread into so many different hosts, including humans, and they mix their genes like decks of cards across species, then I personally don't think that it makes a lot of sense that we look at these viruses in compartments. But we need a different approach. And as I said before, look at this recent pandemic virus. I mean, he is a mosaic. He comes from, he's really a globetrotter. And how can we fight, or better, how can we learn to predict more if we continue to look at these infections in boxes? And it comes back to me, the words that I've heard this morning, fragmentation, fragmentation. We really need to join the dots, especially because these animals that are carrying viruses and products that are carrying viruses, let alone the people that are carrying viruses, are traveling around the world. Now, this here, anybody know what this is? This is the transcontinental routes of meat. So if we want to eat an Argentinian steak, the Argentinian steak has to come from Argentina, okay? Or else you're conned, so it depends what you want. And there's massive circulation of pork meat, of poultry meat from the areas that produce this meat. These are the migratory flyways. Of course, birds carry the viruses. And anybody know what this is? Ships. There's a sailor we have in here. Yeah, so it's ships in the sea. These are the ships in the sea now. If you go on this website, you're able to track all the ships in the sea. And ships carry all sorts of things. They carry people, they carry human clandestines, they carry products, illegal products, legal products, everything. And of course, people have peculiar alimentary habits, and so they carry all sorts of things. I mean, the harbor police or the harbor veterinary services, they continuously seize products, illegal products that are imported from China all the time, and we isolate viruses from them. It happens all the time. So the challenge I have for you is, does it not make sense that in a globalized environment and with all these movements of viruses basically that hitchhike on the back of anything they can find, that we look at influenza infections as one whole, as one problem? And human beings can do extraordinary things. They can not only decide they're going to Mars, they can decide when they're going to Mars, and they can produce, a weather report from Mars. There it is. Which I don't really know how useful that is, but I promise that it does require analyzing a hell of a lot of data. A hell of a lot. A weekly weather report from Mars. And this brings me to the last slides of my talk. A few years ago, I got myself in an absolutely gigantic mess, okay, absolutely, which was much bigger than I am, even if I'm a big girl, but that was a really big mess. But my heart was and is in the right place. So my lab is very active in working with African and Middle Eastern countries. We've heard talks before me talking about how important it is to develop the infrastructure, to help developing countries to build trust. And you know what? Italians, nobody's afraid of Italians. There must be a reason for that. So Italians, we have the credibility we have, but, we're Mediterraneans, and we have a lot of good contacts in Africa and in the Middle East. So basically, our lab was the first lab to characterize H5N1 when it arrived in Africa. And at the time, we're talking about 2006, the genetic characteristics of this virus would have been essential to understand what was happening, because let's not forget that nobody knew what was happening at the time. We didn't know how H5N1 was moving around. I mean, everybody was, wearing masks in front of swans, if you remember. So this would have been something really important for the scientific community. And we were asked to deposit the sequence in a password-protected database, and I said, look, I really don't think that this is reasonable, because the whole world is looking at H5N1, and it doesn't really make sense that only a restricted number of labs can look at this information. This is information that we should all work on together. So I deposited that sequence in GenBank, which was downloaded 1,000 times in a week. And I mean, I really wasn't prepared for what was coming. I wasn't, and this was a start, okay? So, I work in an Italian lab, which isn't the NIH, okay? So we're not used to this sort of attention. But apart from this, I was all over the press. Even speculating on certain things, and saying there's secret archives and blah, blah, blah. But certainly, there was a greater need for transparency, and there still is a greater need for transparency. And the funny thing is that following this, I was actually awarded, as Gigi said, 2 prizes that, for leadership in science, and this, which was a bit scary, for doing something which I thought was quite obvious. Because if you can't do it, you can't do it. I mean, I didn't want to be one of the club. I wanted to make sure that the results of the research that we were generating were used at a global level to make things better in a moment of crisis. And so I think that it's at times worrying how you get awards for doing things that are really obvious. But anyway, apart from this, all this noise and all this mess, has generated changes. It has generated important changes, a lot of irritation, I promise, but changes. Now the World Organization for Animal Health says that any animal reference laboratory who isolates an influenza virus has to deposit the sequence of that virus within 3 months of its isolation in a publicly accessible database. The WHO acknowledges the importance of sharing genetic data, and KG mentioned it before, and we all understand that it's very complicated, but I think, and of course the FAO also promotes the active sharing of information. And so I think that now we are in a different position compared to where we were before. And so the proposal and what I'm trying to move forward is, why don't we take all this information and look at it in a holistic manner, in line with the One Health vision? Certainly, we need to improve our prediction capacities, and I think that everybody agrees with this, given that the pandemic H1N1, I mean, we didn't even go near guessing what was going to come next. We have spent millions, I may say billions, on developing influenza capacity for surveillance. USAID has spent millions, and there are labs out there, human labs and veterinary labs, that are working and that know what they're doing. And if this isn't pushed forward, if we don't find a way to keep this going, this is going to die, because they have other priorities and it's going to finish. We have an increased awareness of the role of the animal reservoir in generating pandemic strains, and sharing and transparency issues are being resolved. They're being worked at, and there is a way forward. There must be a way forward. So what's behind this idea of one flu? It is, as I said, to improve our prediction skills. If we understand how these genes of influenza viruses migrate across species and continents as one evolving gene pool, if we are able to really do human and animal surveillance and research efforts so that things can move forward together, complementing, each area complementing the other, we can maybe be able to identify the determinants for virulence of these viruses, of the next pandemic virus. And we can really work hand in hand trying to push the One Health agenda forward. So in a nutshell, these are the bullet points of how I see this working. Capitalize on investments and networks established for H5N1, which can then be used for many other things, for many other diseases. Perform harmonized and targeted surveillance for humans and animals, and develop and maintain a permanent observatory, a permanent influenza gene pool observatory, which is basically something like a library, which is something where people around the world are in charge of making sure that the information that is generated through these surveillance efforts is collected and is looked at, so that we can, maybe in 20 years, be able to predict better what the next pandemic is going to be caused by and how severe it's going to be, and so on and so forth. And basically, we would work hand in hand for preparedness, transdisciplinary collaboration, control and mitigation strategies, and early warning systems. And most of all, the OIE, WHO, and FAO have put together another document, which was published in April this year, which is the collaboration. And so I think that with influenza, we can really put meat on the bone on showing how this collaboration can work and how this agenda can move forward. And so thanks for your attention. And so I won't get another shot at this, speaking to such a distinguished audience and people that can really change things. So please try and keep these concepts in mind, at least for the next minute and a half. Thanks very much. Yeah.
We have time for a couple of questions if people would like to.
I was fascinated with the statement. Thank you. It's Kay Marish. I'm from Homeland Security. You mentioned H1N1 2009 as swine flu. My question may be philosophical or scientific. Do you have the evidence to support such a statement, knowing that the genetic makeup is avian, human, and swine? Thank you.
Well, it appears that the virus has emerged from swine, and we know that swine have a very peculiar role in the epidemiology and in the ecology of influenza infections. So they are able to be infected with viruses of avian origin, of mammalian origin. And of course, I mean, nobody was there with the swab in the nose of the pig and the swab in the nose of the child. Also because maybe surveillance in Mexico, the human-animal interface maybe isn't as good as it should be. But in any case, I think that we do have evidence that this virus was circulating in pigs. And on top of that, the fact that the virus spilled back into pigs and is continuing to spill back into pigs in many parts of the world, I think that shows that that's, I mean, the best guess that I have, at least, that that's where it came out of. But just to add something on the back of that, had we had a good surveillance program in pigs, you wouldn't be asking that question. The problem is that there are enormous gaps in animal surveillance, and therefore our data is a little bit patchy here and there.
This is Eric Toner from the Center for Biosecurity, and that's exactly my question, which is why don't we have better surveillance in pigs and in birds, poultry and swine, commercial swine? It would seem that there's an obvious disadvantage or disincentive for the farmers to participate in such programs. Can you speak to that?
I can, because I'm Italian, and so won't hopefully be shot when I walk out of this room, because like I had sort of nothing to do with it. But I know the problem. I spent 2 months last year in summer 2009 at the CDC, and I was supposed to work on something different and not on influenza, but of course it wasn't possible. And so one of the problems that we faced, or that was dumped on my desk, was so what are we going to do about the animal surveillance, particularly in pigs in this country? And I was really surprised because, and I was speaking to the delegates of the European Union here about this, and to other people. In Europe, we have a different system, and we have the possibility of sending official veterinarians to swab pigs and sending the results to a public lab and looking at those results. Here it seems to be much more difficult. I think that there's a cultural problem. The food industry is very important and very powerful in this country, but I also think that we have to find a compromise in this. And in fact, the whole idea of doing a gene pool surveillance, right? This is based on a gene pool surveillance, is because the idea behind it originally was if we only identify selected genes of viruses in that population, we won't necessarily have to say it's swine flu. Because you all understand that there are absolutely no food safety issues with swine flu. There should be no trade barriers. So the industry is suffering from unjustified trade barriers, and they're afraid to open their books because they're afraid that they're going to lose more money. And so we as public health officials, we need to find a way to not make them lose money and not make them go bankrupt, but find out what's happening. Because this virus was a mild virus. It could have not been. So I think that we need to find a compromise, which is not going to be easy, I understand. But
Hi, my name is Don Burke, the University of Pittsburgh. I'll be speaking a little later about diagnostics for humans. Could you comment on the state of the art and the utility of serologic diagnoses for serosurveillance in animal populations? It's sometimes very difficult to find the virus at the moment of acute infection, but it leaves behind a footprint in the immune system, which is antibodies. Is that useful now? Is that an area that that would be more useful in veterinary epidemiology?
Well, it depends of what virus and what species you're talking about. I can tell you that doing surveillance for the pandemic virus, for example, in pigs, from a serological point of view, would be of no help. There's vaccines that are being used. There are... Because there's cross-reactivity between the antibodies, there's other H1s that are circulating, and it's very, very difficult. And in some animal species, it can work, like dogs, although I understand, because in the US you have a problem with flu in dogs, but now there is a vaccine, which there wasn't a few years ago, so that seropositivity was just linked to field exposure, and there's one virus circulating in dogs. In pigs, there's many, many influenza viruses. So it can be of some use, but you need to know exactly what you're looking for, and you need to use the appropriate test to give you the answer that you were looking for.
Do you think the development is being done well enough for those aspects?
We're working on it, but the vets are always under-resourced.
Well, thank you very much.
Thank you very much.